All right, so now let's get started. Today, we are continuing to cover the stereotaxic surgeries and in vivo techniques. So I think Shukla did a good job for actually like complementing some of the contents for previous and today's lecture. So stereotaxic frame and surgery, the demo will be done on this Wednesday. So be ready if you really touch, I think you feel the sense. So look at this craniotomy surgery with a drill. And you already know that this is what you will be actually touching in this Wednesday. And you see this numbering 1, 2, 3, 4, 5. And so the actual craniotomy surgery suit looks like this. So you have this stereotaxic device, you put your animal there, and you have also surgical instruments. Of course, you have to sterilize beforehand, and you have a micro drill, which is typically, we use it for a dental clinic for human. And also microscope, it's not absolutely necessary, but this helps a lot because our eye, compared to this surgical microscope, makes it much cleaner and bigger for you to do your surgery, microsurgery. So let me check. Jo Ji-hoon, maybe it's a good time to just give you remind this. So what does this number 1 will be do out of these 3?
Sagittal?
It's a, no. Like 1, 2, 3 axis. Like
What is 0?
choose out of this. This is vertical. Back to belly. Back to belly.
Back to belly. Transverse?
Sorry, you can choose out of these 3.
Sorry. One is anterior-posterior.
Yeah. 1 is.
And 2 is
2 is.
medial lateral.
Oh, that's true. Yeah, 2 is medial lateral, right? 3.
3 is dorsal ventral.
Yeah, 1 and 3 are actually incorrect. Sorry, because it might not be clear to you. So the animal looks like here. 1 is ups and down, so it is actually dorsal ventral. And 3 is like frontal backside. This arrangement, that is anterior posterior. Is it clear, Ji-hoon?
Yeah.
Okay, yes. All right. So today, the objective is we continue to talk about common implants that will allow temporary or permanent access to the brain. And we will have an extensive session later on about how to measure neural activity. But here we will cover briefly measuring brain activity and neurochemistry in vivo. So what techniques we will be covering? So last time we talked about surgeries in general, and now we will be a little more specific about implants that will allow access to the animal brain. So for that, we'll talk about C-level chambers and cannula and cranial windows, which you will be, last demo, you will be actually performing mouse brain surgery yourself to install cranial windows. And the thirdly, measuring neural activity. Measure neural activity there are 2 big ways. One is electrophysiology, and the second is in imaging. So for example, fluorescence activity indicators such as calcium imaging. And the last one is measuring neurochemistry. We will be talking about microdialysis and photometry, and part of it as an amperometry. All right, so why do we need to implant for long-term access to the brain? And we sometimes put implantable devices, which allows us to have a long-term access to the brain for chronic injections or physiological recordings. And these three we'll be talking about. So first, C-level chambers. And the left one is, in fact, this figure is for monkey, okay? So when we study monkey neural activity by imaging, we actually use this one sometime. So you can see the skull is gone over here. So by using drill, we remove the skull, part of the skull of a specific brain region of our interest. And then you can see these dark dark lines are actually durometer, which Shukla nicely tell you that it's like very sturdy one, which covers the brain, and inside there's a CSF. So it's like, as I said, latex rubber like one, but in mouse, this is very, very thin. It's even hard to see, but in the rat, this thickness is a little more thicker, so it can interfere for deep tissue imaging, okay? So in this monkey case, dura will be actually pretty thick for easy use for optical imaging. So that's why we remove the durometer and we put these implantable, transparent, they name it as artificial dura. And what's cool thing about here is there's a cap here, so that it means you can put it there or you can remove, so that when you have to inject something or you have to measure electrophysiology by inserting electrode, you can actually use these C-level chambers. So around hollow with one end affixed to the skull and the other end kept with a screw top. But in mouse is so small that you don't usually have this one. Then the question is, you know, like when we talk about central nervous system, including brain and spinal cord, then people will be also asking about why not spine? Can we see spine in microscopic detail in vivo? The answer is yes. People just have created a permanent window on the spine, and that has come out in 2012 Nature Methods by Chris Sheffer's group, who is in Cornell University. So that's in the right-hand side. You can see you have a mechanical drawing skill. You can create this a spinal window. So look at this spinal cord, and you can take off some bones which is on top of the spinal cord in this part, and you can remove that. You see this red blood vessel, and then you can create a mechanical device like this. You see it's 10 millimeters, so it's a pretty small, tiny, and you can do this surgery on the mouse spinal cord, and that can last long. So this paper in Nature Methods, you actually can look at when you create this a new model, what to do with this. So you want to show for first week, second week, several weeks, how clean the window qualities are. And you can use a advanced microscopy like 2-photon microscope, actually watch cellular level blood vessel, and there are neurons and also glial cells. You can watch them over and over time, and this allows us to study, for example, when we study a pain, you can have a pain model by like directly hitting the spinal cord, giving a damage, and how this damage affect, such as a at the cellular level of, for example, microglial activation, or there's a glial satellite cells. How does this cell distribution are different? Also, even you can actually insert microelectrode and to measure electrophysiological changes. So because you know, back pain is many elderly people have it, right? My both parents have gone through surgeries in spinal cord. So this is also very, very important at the microscopic scale to study. All right, and another thing to directly access inside central nervous system, we actually have to have an access, but there are always bones, skull, like bones are there. So we want to put a narrow, a cylindrical, in fact, it's tube. So this is called cannula, can be inserted into the brain, and we can permanently affix to the skull. You see there are like glues and cement to fix it, and to access deep brain structures within the brain. So for example, you can see here in this deep region of the brain, you actually insert. And you can not only insert one cannula, but you can insert multiple. So in that case, this plural is called, you can see in the title, cannula. Okay, so why do we need this? To deliver substances or probes through. So for example, we also want to give a drug, so pharmacological agents, or part of the brain in specific region. I want to inject a vital vectors. For example, I want to have a calcium indicator in specific neurons in the brain, such as this area. The virus does a good job for infecting cells so that we can insert neuron-specific, let's say, activity indicator or fluorescence indicator together, and then we inject, and later on you can check and you can do imaging, you can do electrophysiology. Or you can also insert narrow electrode. You can see, you can put electrode anywhere inside the brain so that you can measure the electrical activity of the brain. Okay. And more cool way is even optical probe you can insert. And nowadays a new technique, a relatively new technique called a like optogenetics, or you can measure this calcium imaging indicator activity, not only the surface of the brain, but inside the brain by using optical fiber probes. So these cannula can be made of plastic, glass, or steel with various sizes. The important thing is, like, this is a quite delicate experiment, and at the end of the experiment, how do you know that what you injected or inserted is the right position? So you need to verify for yourself and for your publication as well. So correct placement of cannula by examination of brain sections. Let's say, Hanjinu, can you guess what this is?
Histological.
Exactly, yes, right. You always have to prove yourself after finishing, then you fix the brain and cut it, and then show that the location you injected is in the, in fact, the right place. Okay, so now I want to discuss about cranial windows. This left top one is actually, I made this window when I was a postdoc. This is a nude mouse with a cranial window. In fact, that in the brain, there's a tumors growing in the brain. And this is actually very primitive and crude approach, because you see, this is not stereotaxic device. There's like just this L shape. You just put it under the animal, and I put some sponge here so that it reduces maybe pain for the animal. But you have to adjust the tilt by hands and with tooth bar here. But with stereotaxic device, you actually need sometimes a better control. To make this window flat is actually very challenging things. So let's look at the bottom figure. Sorry, I updated this slide. I put it in Basecamp. So you see there's a microscope objective. Those who have used microscope with this 20x, pretty high magnification, and you will find the working distance is actually not very long, such as only 1-2 millimeter away. So you have this bone, you have a craniotomy, and you have to have something to replace the skull, which is transparent. So for example, you can have an optical window, but you have to fix it So you need to use a specific glue. For example, you can mix with a crazy glue with a powder, such as a nail polish, or nowadays more of a dental cement. We are using it so that underlying neurons you can see. So the issue here is to visualize proofs in vivo, we need an observation window through the skull, and that for long terms of time. So how long does this window can last? You know, it depends on the surgical skills and sterility of your surgical environment. So depending on your purpose, you can observe it on the acutely, just right after surgery, you can actually do experiment and maybe finishing experiment. But many times, you also want to do long-term study or longitudinal study. Then you have to make sure your surgery is very well controlled and can last for weeks or even years. You know, mouse lives up to about 2 years. So a very good window can last until the mouse dies. So this is a very difficult skill to develop, but I want you to experience this. You know, learning is one day, but to perfect can take several months to years, okay? So let's see. Here, let's see. So we need a cranial, this I'm sure everyone knows craniotomy, because you have to cut the cranium bone and replace it with a glass cover slip. And the important thing is we are talking about a microscopy. So this is one type of a microscopy, but you need to put a really fixed head when you do microscopic experiment. The important thing is it depends on the target, such as you want to watch blood vessel, big blood vessel, maybe this is okay to use a crude one such as this. It's fine. But let's say you want to image a single neuron is fine. That is an order of 1 micron. Or I was usually interested in observing microvasculature such as capillary, which has a diameter of about 4 micron diameter. This is tiny, tiny, and you use this, forget about it. You can't image. So in that case, you need a better fixation, really sturdy. You have to think like a mechanical engineer to consider how to make it. Okay, so you need to implant sometimes head post for proper alignment. I give an example here. So you give a metal plate using titanium. You implant this in addition to the window, and then you have these bolts and tools to really fix the brain of the animal of interest during your imaging. So another aspect is because of this optical window, sometimes we can't interact underneath the neuron, or you can't measure it directly. So there's an invention that why not using something flexible and transparent, and like a transparent plastic icon, such as called PDMS. So here it is. This is developed by Professor Samina from Sungkyunkwan University. So this was in 2016, that you can actually have a flexible PDMS instead of a window. And this resin and PDMS allows you to actually insert a needle so that maybe you can inject tumors, or you can inject calcium indicators. And this is a good example of a red craniotomy. And you see how to do this experiment. 1 week up to 10 weeks, you demonstrate that the quality of the window is still clean. This is quite rare though. So when you do a good job, this is possible. Okay, so in fact, I have brought some of the window myself. So let me share the screen. Let's see. Okay. So can you see my screen? Okay, so let me show some of the examples here. So this is the mouse skull I've made. So this is tiny 3 millimeter diameter, very, very thin. It's only about hundreds of micron thickness. So these are an example. So after that, you can use this angel ring. This one, you put it on the surrounding window to grab it. How we grab it? So this is an example too. Actually, this device is made in South Korea for the LCI company. So you have these.
Sorry, Professor, screen is stopped.
Oh, really? Okay. Okay, sorry. Then I think it's better to show you with this window. Okay, have you seen this? Can you see this in the ring? You have this ring, right? Now it's clean to you, right? Oh, please answer. Okay, raise your hands. Can you see it? Okay, good. And to fix this, we have these tools. So you can have so you have angel ring and you have you can use this one to lock. Okay. So this is for the mouse, and here this side is for gas anesthesia. So, and also this plate is connected to heating pad, so it contains a heating pad. So make sure when you are imaging all these, you make sure the animal is not under hypothermia. Make the body temperature heating so that to keep the physiology. And another example I brought is these. So these are also called head bar. So how do you use it? So you have this, let's say this is mouse brain and window, you put the head bar, get stuck onto, fixed to the skull so that later on you actually use this head bar to grab it during the imaging. And this sometimes is not strong enough. So you create these ones. You see the little hinge here. So to make sure when you attach onto the skull, make sure it cleans. And even you can see, and I don't know, you can see, I grind this part, which is, this is for the shape of the mouse skull is curved. So this way you can attach it more closely so that you can grab it better. And all these head bars are made out of titanium. So that it's a lightweight but strong enough. Okay. So, all right. Hopefully this gives you some idea. Let me change my screen. Okay. All right. Okay, thank you. All right, so chronic cranial window and thin skull preparation. This is a reference, but I want to share some of the history in cranial window. So by using this cranial window, people have done a lot of interesting studies. For example, so in the neurons, spinal neurons, dendrite has lots of synapses. So in this neuron contains this synaptic junction we call as a spinal spine, dendritic spine. The size is only about 1 micron, very, very tiny. And people realize that like these spines are like sometimes making up or disappearing and changing to shape, volume, size. So we consider this as a, hey, the neurons are not fixed. Neuron is plastic. Our brain, when we use it, or when we are uh, like memorizing, this activity makes the spine, dendritic spine changes, and we can observe it with the microscope over time. So for example, you have a small mouse, you block one eye, and then the corresponding visual cortex of this left and the right, the spinal changes you can observe over a long time. And people have done this experiment to realize we can have a microscopic scale to understand the neuronal level plasticity, which is exciting. So some people have made the spine turn over time, and then they publish in Nature, okay? And later on, some people realize, hey, what's the limitation of this? The limitation is that you see scar when we like grind the scar. You see these are meningeal layer, which is composed of 3 layers. Hi, you know. And by doing this surgery itself will inevitably inflame and damage this meningeal layer, such as dura mater, arachnoid mater, and pia mater. So another idea is that, hey, why don't we not to try to preserve this meninges? And then how do we do it? We just grind and make the scar thin enough. So it's very thin, then it can be transparent. I have to say it's not perfect, but it may be good enough for a shallow observation with a microscope, okay? So this way you can thin the scar, make this meninge or dura intact, and people have measured the turnover time of spines, dendritic spine. They realize the turnover times different. So they publish in Nature Neuroscience, hey, the previous study with this total open, we call open cranial window, maybe not physiologically relevant. So these are the debates has been done. So now people do both. If you want very physiologically relevant, and that can be certain to be related to this inflammation and damage to the dura, you may want to go to this thin scar while your image quality and imaging depth will be compromised. All right. So after stereotaxic surgeries, neural activity can be measured in vivo in 2 ways. One is electrophysiology, and the second is visualization, especially fluorescent proteins. So electrophysiology is recalled the electrical activity of neurons, which will be covered next week topic. Visualization of fluorescent proteins, and we use like 2 ways. One is fluorescent dyes. Dyes are usually super bright, but these are exogenous agents. And the other is genetically targeted activity, which is usually using transgenic animals, genetic targeted activity sensors. Or you can have a viral vectors to deliver, such as calcium indicators, into the target neuron population so that you can use fluorescence imaging to measure the neuronal activity. But these require cranial windows, so that's why this is so important. So then let's see about electrodes for electrophysiology. So here I showed some, this is like wire, electric wire, like you can use a tungsten. And these wires as a single electrode, that's often mounted on a, when you are getting into the brain, let's say mouse or rat, you're talking a very, very tiny displacement. So you can't use your hand to target. So you typically use micro drive to precisely raise or lower that electrode to specific depth inside the brain. And but single mic, this electrode is mechanically not that stable. It can bend easily, So people have made like 4 of this to make it more sturdy. So you can see that as a type of multi-electrode array consisting of 4 active electrodes you can see here. And this is still narrow enough and can be easily inserted into relatively deep brain structure, because a single electrode, a single wire might not be strong enough to target deep part. Okay, let's see. So, 지범. Okay, so now when we are watching only one or a few, but there are like hundreds and thousands of neurons to probe. So we may not be satisfied with this approach. So we may create a grid of dozens of electrodes, such as 8 by 8, that is 64 electrodes, or nowadays so-called high density, which is thousands of electrodes to record the activity of multiple neurons nearly surface of the brain, usually outer surface of the brain. Jibam, can you guess what this is? I can't hear you. Oh, it doesn't work. Okay, that's fine. So it's a multi-electrode arrays. So one example here is a so-called, I added this because it's so famous, called Utah array. So this we call multi-electrode array, or MEA. And this is Utah in vivo electrode, right? You can see like 8 by 8, 64 channels you can measure. But I'm showing in vivo, but in fact, MEA has an in vitro version is probably more popular. So that version, you do not have these pins, but you can grow neurons in culture, and multiple neurons can be measured by using this MEA as a substrate. Okay, just to give you an idea of that. Then let's think about what's happening when you insert an electrode inside the brain. So for example, type of these 4 active electrodes. Let's see, I think. Yanguna, so I guess you can tell what this 4 type of electrode?
Tetrode.
Yes, exactly. So it's 4 is tetrode. And the tetrode is one part, you have 4. But sometimes, for example, many of the cortex are layered structure. So we sometimes, for example, mouse has 1 layer, 2, 3, 4, 5. So these multi-layer you want to probe, then you can have this different probe height, and you insert, and you can measure like deep structure from layer to layer electrical activity. So let's see what's happening when you insert it. Let's say tetrode, a size is about like several microns. So these are a few micron, and neurons, you know, cell body is about the order of 10 micron, but you see there are like hundreds of neurons. And within the tetrode, you can think of the electrical activity of these neurons. For example, this neuron generates electrical activity, and that can be measured in different parts. But depending on different distance, the signal has a different time lag. And also different neurons will generate different signals, either simultaneously. Then what you get, these electrical signals, now you have to solve the problem of these signals. Different neurons have different shapes depending on the geometry and their activity, so that you can actually like decompose this electrical signal and figure out how many neurons are nearby to detect. So that we call spike sorting or clustering, because these are like example 2 neurons electrical signals. Okay, hopefully this makes some good sense to you. All right, so now I'm gonna talk about neurochemistry in vivo. So to know specific neurochemical changes in over time. So let's say, I think 고윤해?
I'm not sure.
Yeah, it's a concentration over time, okay? So there are many neurochemicals inside the brain, such as neurotransmitters. And the problem is this is not very easy technique to run. So because like in vivo measurement is not that easy. In vitro, there are more sensitive assays are available. So what we do is in neurochemical expression and in discrete brain regions, specific brain regions, and when there's a specific behavior, okay, or physiological function, let's say giving the animal, let's say some like starter, like giving fear, then you want to check specific neurochemicals before and after. So that by measuring a specific neurochemical and you try to link behavior or certain physiology, what you do is not usually absolute concentration, but more of a changes. So relative changes. And Yune, can you guess what this? You want to connect changes in neurochemical expression with the specific behaviors. Yeah, this similar. Yes, it's correlate. So it's like very difficult task. You first establish correlation first, and then you can go more specific and complicated. So what are we interested to measure? Neurotransmitters, hormones, or neuropeptides? And how we do measure this? This is a tough technique, and there are tools for that. So have you heard dialysis, right? Microdialysis and photometry. So, you know, voltage, okay? So this is to identify and measure neurochemicals in vivo. I mean, in vivo technique is hard, but it gives a sometimes more precious information. Okay, so let's talk about microdialysis. So here, this is mouse, and you have targeting a specific region of the brain of your interest, and then you send out so-called perfusate. You pumped in, and then it actually coming out. That's called dialysate. That's pumped out of the brain, which contains neurochemicals of your interest. Because of this shape, it has this Y shape, and you see this probe is all Y shape. Okay, so what are we trying to measure here? To sample the substances from the extra 유승열. Can you guess what this is? Extra fluid in a specific location in the brain? Oh, I can't hear you. Yes, okay, just write it down. Okay, that's fine. Yeah, you're not talking about inside the cells or neurons. When you just insert it, most of the time it's extracellular, okay? And important thing is this microdialysis probe actually create a concentration gradient because you are keep injecting a perfusate into the extracellular, so fluid in the probed brain region, and you are continuously perfusing a physiological solution such as a saline or artificial CSF. So what would be the physical principle? Sunyoung, again, can you guess what this is? You have a physics major, right? Yeah, it's a diffusion. Okay, so let's go next step so that we can see it more clearly. So the principles of microdialysis is you see this Y shape. There is a inlet and there's an outlet, and with the shaft, and there is a membrane. So look at this more closely. So you can constantly perfuse with this perfusate, which is shown as a white ball shape, and then it goes. And you see this dotted line, it's a semi-permeable membrane. And because of this, you see this black triangle can actually getting into, and that comes out as a dialysate contains, and I think I don't have it, it's analyte, okay? This is analyte. So this is based on the diffusion, and so microdialysis probe uses a semi-permeable membrane, which is connected to two thin cannulas. So it's cannula, and we are talking about due to concentration. Let's say Minson. Yeah.
Due to the concentric gradient.
Exactly, yes. Because of the differences of concentration gradient of this analyte, the substance present in extracellular fluid, we are talking a very, very slow process to not disturb the physiology. So it's passive diffusion through the probe's membrane into the collected solution. And the collected solution is called dialysate. Okay, the answer is usually nearby. And then you collect this one for now. You use this contains this analyte of your interest. So sensitive chemical method, now you follow up with high pressure liquid chromatography, radio immunoassay, or mass spectroscopy, so that you can measure this tiny but very sensitive detection of neurochemical. You look at that, this is very, very, very slow, okay? 0.3 microliter per minute to allow passive diffusion to happen. So with this, we can see that some of the features or characteristics of microdialysis. The cool thing is you can directly measure neurochemicals in the brain. And the collection of extra, Sunyoung already told, a cellular fluid can be used for sensitive chemical assay follow-up to identify small molecules. And this microdialysis, now you make a hole in the brain to directly access so that you can be combined with extracellular electrophysiology. You can insert electrode simultaneously so you can match and correlate, or you can even use electrical stimulation, even in freely moving animals. And now you have a hole in the brain, you target the region, you have a connection. And so far when I say microdialysis, is you are trying to get these chemicals which is inside the brain, the neurochemicals, to collect them. Now, because you have a route, you can actually instead, you can give some stuff into that part, okay? So you can deliver drugs into the perfusion fluid. Then instead of like injecting with a, like say, syringe into the brain with a bolus injection, that would not be very good in terms of physiological. But this dialysis is so slow and passive, so gentle, you can have a better control over flow of the pharmacological agent compared to a pressure injection. And Yang Euna, can you guess what this is?
Sorry, not sure.
Okay, yeah, this is just the opposite ways of microdialysis. So it's reverse microdialysis. Because original microdialysis is you just collect, now you give them a drug, okay? And so far so good. So what are the limitations? It has it takes too long time, so long sampling period, about tens of minutes, which may not be good for detecting neurotransmitters released at a physiological time scale, because we know neuronal activity, the time scale is a millisecond, tens of millisecond order, okay? but it takes minutes to order, so that we are more interested in long-term change in neurochemical concentration and may not be a real-time analysis. So that disadvantage is low temporal resolution. Okay, so now let's change gears a little bit to offer another ways of neurochemistry, which is called photometry. So this is a electroanalytical method used in analytic chemistry. So we want to know about analyte by measuring not the chemistry itself, but the current as a function of voltage. So the schematic is here. You have a working electrode, which is inserted into your interest or your interest in reference and counter electrode. So you apply voltage and you measure the current, okay? That's called photometry. So this technique to detect neurochemicals capable of undergoing, now you are talking about the neurochemicals which has a electroactive molecule. And usually the reaction is you give a positive potential voltage and you suck this negative charge electron. So loss of electron, it's called oxidation. Okay, so for example, neurotransmitters such as serotonin, catecholamines, epinephrine, norepinephrine, dopamine, and these are some of the neurochemicals you can probe with photometry. And how you insert this carbon fiber microelectrode into the brain and apply specific voltage. Okay, so how do we do this? So over time, in fact, this is a you are changing the applied voltage. So you see, from low to high, it makes it positive. So you suck, taking out the electron to oxidize these molecules. So it's oxidation and then reduction. And while doing this, what you are measuring is current. So this is VI diagram. So this, when you apply voltage, like from -0.4 to +1, and then you go back to this reduction, then these are of current and voltage diagram, but there are lots of noise involved. So you have to subtract a background, and then you get, this is what you get. So this looks like a so-called hysteresis diagram. So the order is like this: oxidation and reduction. And that can be a little fast. So this can be done within 1 second, so that you see, you even give a stimulus and you can measure the current. So this E is the final diagram I want to show. It's maybe not that clean, but you see, voltometry is you set up specific potential voltage and you measure the amount of current, which is based on this scale bar as a color, and you can do over time. And this can be correlated with the stimulus time, and you see the changes happening here. So this is giving information about specific electroactive neurochemicals. So analyzing and depicting the cyclic photometry. Oh, because of this nature, we call it cyclic photometry. So when the chemical encounters the surface of electron, they undergo an oxidation reaction by releasing electrons while you do this, increasing the potential. That electrons that produce a measurable change, is in current, okay? The charged particle flow is current. And the magnitude of the current is proportional to the number of molecules oxidized neurochemical at a physiological time scale. Now the good thing is we can be a little more faster than microdialysis, but this is an indirect method. So I want to cover a little bit further for so-called exposing you fast to scan cyclic photometry to make it a fast, high temporal resolution And that voltage of an electrode sweeps from non-oxidizing potential to an oxidizing potential within a millisecond. So what you see, cyclic photometry, is to plot the amount of measured current versus applied voltage, which I think here, measured current and applied voltage. And we want to identify specific neurotransmitters as the electroactive neurochemicals are oxidized and reduced at different potentials. So by this shape, you can identify this specific neurotransmitter, neurochemical. And one subtype of photometry is called a, current unit is ampere. So this is amperometry. And now this is a little bit different from the cyclic photometry in that because it holds electrode at specific and constant voltage, unlike fast cyclic photometry, which like sweeps the voltage and measure the current. Now on amperometry, you fix the voltage. Then all molecules that can be oxidized at that potential will be detected, okay? And what's good about this, and you can average it over a long time because you're not sweeping, so more precise measurement of relative concentration of neurochemicals. However, because you have a specific constant voltage, all the molecules that can be oxidized will be detected. So it's a little difficult to identify specific compound detected by this electrodes solely based on the current itself. Okay, so that's, this is the last one. So we covered about this measuring neurochemicals and accessing the brain by using implantable chambers. And now, once we've done this, now it's time to manipulate neural activity in vivo. So how to determine the role of brain regions and to correlate with animal behavior. And we need the tools and method to penetrate the brain and stereotaxically target specific brain region. And with that, hey, I want to, let's say, maybe inactivate or make a lesion, okay, of the specific region of the brain to determine if that brain region is for that particular behavior to occur. That's called loss of function experiment. Min-sang, can you guess what this is? Hey, let's say, I'll give you an example. Hey, I target a specific brain region, for example, hippocampus. I inject a toxin to destroy hippocampus. And then I realize I cannot remember like memory. I lost my memory. So that means that part of the brain region with my memory, this is called loss of function experiment. So that means that brain region of hippocampus is necessary or sufficient for that memorization behavior to occur. Yes, exactly. It's about necessity. On the other hand, there are gain of function experiments and you stimulate neural activity. And then let's say, this is a hypothetical. Let's say you have a neuromodulation of a specific brain region, and then you realize the memory boosts up, okay? Then you can conclude that that boosting up of the memory is by stimulating that particular region is sufficient to cause a behavior. So you want to have these two aspects of manipulation of brain activity. And for these, a diverse toolkit of physical, pharmacological, and electrical methods with the also nowadays more cutting edge genetically encoded neural actuators such as a calcium indicator or voltage sensitive dye can be utilized. Okay, let's summarize today's lecture. So we talked about stereotaxic frame and surgery. Hopefully this Wednesday, you guys all have enough chance to experience, there's a very small little like homeworks for you. You have to take what you did and like 3 pictures I heard, and then you have to write down within a page. So not a lot, but make sure you understand and to obtain what's supposed to do. And we talk about implants for long-term access. So typically when you do the surgery, roughly, maybe the window sometimes cannot last long, like even may not last several weeks, okay? So that implant, we can have a chamber, or if we are not using imaging, you can put a cannula, or for imaging, you can have a cranial window. Measuring activities in vivo, we can use electrophysiology or fluorescence indicators. And measuring neurochemistry in vivo, you can use microdialysis and photometry. I'm not talking about these are very easy. This is some technique that you sometimes you hold your master thesis or your PhD or your like, you know, postdoctoral experience. I'm not talking about that these are easy, but giving to know, depending on what your purpose, you can actually choose to use. And ultimately, we want to manipulate the brain. We know enough, and you can manipulate, you can engineer, and you can actually make a specific behavior or improvement. All right, so that's all for today, and electrophysiology will be covered next week. So thank you for your attention. And let's